In this video, our aim is to convince you that our project is worthy of a $100,000 investment that could boost this research and fast-track it to clinical trials. Prostate cancer is the third most commonly diagnosed cancer in Australia, and age is the most significant risk factor, with 1 in every 5 Australian men being diagnosed with prostate cancer by the age of 85. As our population ages, the impact of prostate cancer is only likely to increase. Prostate cancer is an endocrine-related cancer that develops when rates of cell division and cell death are no longer equal, leading to uncontrollable tumor growth. The epithelial cell layer begins to change, which promotes crosstalk with surrounding stroma, eliciting a mild inflammatory response. The prostatic intraepithelial neoplasia is the precursor to this, and invades the surrounding stroma, blood, and lymphatics, thereby providing a route to distal sites. Patients whose cancer is detected in the early stage of the disease benefit from a high 5-year survival rate due to a range of treatment options such as surgical prostatectomy, radiotherapy and/or chemotherapy, and androgen deprivation therapy. Yet the clinical reality is that most men do present with a more advanced stage of the disease. In the case of metastatic castrate-resistant prostate cancer, the treatments are usually palliative in nature. Recent evidence has implicated the PI3K, AKT, mTOR signaling axis in the development and maintenance of castration-resistant prostate cancer. This pathway integrates growth signals with downstream cellular processes such as protein synthesis, proliferation, survival, metabolism, and differentiation. Hence, this provides the necessary conditions for cancer cells to essentially resist the stress associated with androgen deprivation.
Hi, my name is Jared. I'll be demonstrating our lab's proposed novel therapy to combat prostate cancer by combating 2 hallmarks of cancer, sustaining proliferative cell signaling and activating invasion and metastasis. The retinoic acid receptor-related orphan receptor gamma, or ROR gamma for short, is a part of the nuclear receptor subfamily present in the thymus, lung, liver, kidney, muscle, and more recently in prostate cancer cells. ROR gamma has been a hot topic in recent immunological studies which exhibit effects on regulation of T helper 17 cells. More recently, last year, Junjiang Wang and his team found ROR gamma plays an essential role in androgen receptor expression. Junjiang's work shows through immunoblotting of 4 patients of each group, non-malignant, CRPC, and androgen receptor negative, which results in ROR gamma not being expressed in non-malignant tumors, overly expressed in CRPC, and low amounts of ROR gamma in androgen receptor negative tumors. Highlighting ROR gamma has a crucial role for the progression of prostate cancer into its castrate-resistant form. So how does ROR gamma work? Now, ROR gamma is controlled by the ROR-C gene. Upon activation from a ligand, usually a cholesterol precursor, most commonly desmosterol, leads to ROR gamma binding to DNA with a ROR gamma-specific antigen receptor ROR response element motif known as ROR-E. ROR-E is in the first exon only 2.3 kilobases downstream of the antigen receptor transcription site, but this is not the only signal. ROR gamma also recruits nuclear receptor coactivator 1 and 3, known as SRC1 and SRC3, which also weakly bind to ROR-E to stabilize the connection. Once connection has established to ROR-E, this stimulates antigen receptor and antigen receptor variant 7 genes transcription within the nucleus to promote antigen receptor expression on the cell surface. By engineering a ROR gamma antagonizing chemical structure, let's call it chemical X for now, similar to cholesterol precursors, we hope to create a much-needed therapy for such an advanced stage of prostate cancer. If we were to be successful, ROR-C would still signal, ROR gamma to be expressed, however chemical X now in the environment would bind to ROR gamma, preventing any agonist to bind, resulting in no recruitment of SRC1 or 3, therefore no activation of ROR-E, and ultimately preventing overexpression of androgen receptor on the cell surface. Using chemical X, we hope to combat cell proliferation in the prostate and the lethal hallmarks of metastasis. Current advances in the field of nanomedicine will allow us to directly target drugs to the cells of interest. First, we intend to encapsulate chemical X into small unilamellar liposomes. Next, we'll pegylate our liposomes such that polyethylene glycol polymers are covalently linked to the liposome in order to make the molecule more soluble in the blood, as stated in Research 2015 published by the Journal of Human Gene Therapy. Studies published in 2014 in the journal Molecular Therapy suggested that aptamers are more suitable to target liposomes to specific cells as opposed to antibodies due to their small molecular weight and that they are completely non-immunogenic. A paper published in 2016 in the Journal of Nanomedicine suggests that SZTI01 aptamers bind specifically to prostate-specific membrane antigen found in more than 50% of recurrent prostate cancer cells and almost all metastasis to bone and lymph nodes. As such, we intend to conjugate SZTI01 DNA aptamers to the distal end of our polyethylene glycol polymer, which will target prostate cancer cells. The nanoparticle drug will be injected into the patient intravenously and leave the circulatory system at sites of the tumor via leaky cancer cells. Once in the cancer microenvironment, the liposomes will bind to the cells by SZTI01 prostate antigen linkage. The Liposomes will then be internalized and degraded, releasing chemical X into the cell.
The effect of ROR gamma antagonists on androgen receptor regulated gene programs in CRPC cells appears to be extensive, encompassing androgen receptor targets that are upregulated, downregulated, or persistently expressed in CRPC tumors. However, the overall impact on CRPC tumors is unlikely to be limited only to the androgen receptor. We hope to be at the forefront of developing more potent and orally bioavailable ROR gamma antagonists.